Let me welcome Maria Smith from Proveris. She's going to talk about the guide to the effects of product development and on nasal spray performance. Welcome, Maria.
Thank you. All right, so thank you to Dr. Crespi for asking me to speak today. I am going to go through the guide to effects of product development on nasal spray performance. So this is really pulling all of that information together of how we can actually make this into a product. Just a quick overview. We'll start with who Proveris Scientific is. So basically, where is all this coming from and why are we telling you this? We'll go through then the complexity of nasal spray development, followed by why we believe that early product characterization is so important. Where does human realistic testing come into play and why? And how do formulation and device changes affect the performance? All right, so we'll start with who we are. We have 25 years of experience developing analytical instrumentation for OINDPs, including nasals, 15 years providing laboratory services. We have our regulatory expertise. Dina Farino, our founder and CEO, contributed to developing non-impaction laser imaging and automated actuation technology for the industry and regulatory bodies. We have a global presence covering full range of innovative generic pharmaceutical companies, CROs, CMOs, device manufacturers, academia, and regulatory agencies. We support R&D, NDA, ANDA submissions, quality control, as well as post-approval changes. We base ourselves around a scientific focus. So we have experienced scientists and engineers that are focused on advancing aerosol and spray products, quality, and characterization technologies. So getting into nasal spray product development, there's a lot of complexity involved. You would like to just think it's a formulation, you throw it into a device and you can get it to go where you want it to go. Julie's going to go into a little bit more about that later with the different devices, but there is a lot of complexity. It starts with your API and your formulation, which has to do with your viscosity, surface tension, your thixotropic properties, mucoadhesives, as well as your penetration enhancers. Then it comes into your device. You could have a metering pump, your actuator tip, your bottle and dip tube, the assembly variation, spray geometry, particle size, as well as the proper dose. One of the things then that I think most of you will appreciate probably the most, but a lot of people seem to forget, is the patient administration. So how does the patient use it? What demographics use it? When they're actuating, what force are they using? How are they getting the stroke length to work? Are they holding it long enough? Are they shooting it fast enough? How do they have their head positioned? How do they have the bottle positioned? And how far into the nasal cavity is it going? All of this comes into play with how the product is actually going to perform. And then we also have successful delivery of the API to the target locations, right? So deposition in various respiratory regions, mucociliary escalator clearance, high humidity, constant temperature, and inhaled flow. And all of these can really affect how your product works. So kind of going into what I was speaking about just on the last page, we look at this as a three-legged stool. For the stool to be able to stand up, for your product to be able to work, all three must equally support. If you have any one that's different than the others, it's going to fall over. Your product is not going to work as intended. So for these, you're looking at your formulation, the device that you're going to use, as well as how the user is going to use it. Why early characterization? Right? So as far as the FDA is concerned, spray characterization is not required in phase 1 development. However, if you do the characterization early on, you're going to help to de-risk your process. It will assist in understanding your device formulation relationship. The data can assist in the next development stages. Spray pattern is very sensitive to changes in formulation, device, or actuation. So again, this is really going to help you with trying to get your product where you want it to go. And then your deposition assists with the targeted delivery. So why is it important to translate human actuation into automated actuation? If you read the FDA guidance, they are looking for these products to be tested consistently every time. And if you look at the data that we have on the page here, the manual actuation introduces variability in testing. There are very few devices out there that can be shot the same way by every single person. And excuse me, the graph below here just shows manual actuation versus automated to show how you're able to streamline that and ensure that you have precise control and you eliminate that variability. This is going to help you with your testing so that you know any variability that you see, where it's coming from, or rather where it's not coming from. Your stroke length, when you set that, affects your shot weight. So basically your spray content uniformity. So you can see here, as the stroke length increases, your metered shot weight will also increase, obviously only up until what you have filled the unit to, right? Eventually you are going to hit an end. But generally, if you increase your stroke length, you will get more out of the pump. Velocity affects something different. It affects the actual performance of how it comes out of the device. So this will affect your spray pattern, your plume geometry, and your droplet size distribution. So we have a couple things to show here. Hopefully this works. This shot here is a nasal spray pump. It's a multi-dose nasal spray pump with low velocity. This is the exact same pump when we shoot it a little faster. And again, the exact same with even a higher velocity. So you can see on how the method that you use will drastically change the performance. So now when you think about somebody shooting it with their hand, if you have, say, a child shoot it versus a strong adult, you could get a completely different performance profile out of your device for the exact same product. Okay. Now, this is some data that we have here that shows varying velocities along with varying viscosities, right? So this just shows how you can have across the top here, we have 0%. This is glycerol that we use. So 0% up to 80%. And in using the same method across the top, a velocity of 70 millimeters per second and an acceleration of 5,000 millimeters per second squared, you can see how using the exact same method, you get very different results as your viscosity gets higher. What you can see then, if you jump to the lower section, then now we're using a different method, but again, across the line of those same increasing viscosities. So what this slide is meant to show here is that there is no one size fits all answer for testing these products. Each product needs to individually be developed to find the proper method to test to ensure that it's being used properly as well as being tested properly. This slide here is just going through life testing. So this ensures consistent dose delivery. Again, this is a multi-dose device. During product development, this helps to look at priming and repriming, dose consistency, as well as confirms the doses per bottle. So basically what this is data from 10 devices. It's 1,500 data points per device, so 15,000 data points in total but shows whether or not the device is consistent and whether you can get that same dose across the bottle. If you look all the way at the end, you will see the tail off. And again, this shows your end of life. So basically how many doses are in the bottle. Next, we'll talk about force to actuate measurements. So here we do talk about unit dose versus a multi-dose. As far as the agency is concerned right now, they really only look for this for unit dose, but that doesn't mean that they won't change their mind. But basically looking at this, the force to actuate, in the case of this specific unit dose device, it is the force needed to make the unit fire. So again, it's not always the highest force. As you can see, that force raises along the actuation, but it is the force needed to progress the unit to actually expel. The multi-dose device is the same theory, but that is more of a spike at the very beginning to push through actuation. And the force to actuate, again, comes in a lot when you're talking about your demographics. If you need a child to actuate it, or your patients are elderly, the force to actuate can be very important, because if the force is too high, that patient might not be able to actuate the unit properly to get the product to go where we're trying to get it to go. Okay, moving forward, there is a changing nasal landscape happening around us. And this is kind of what we've been talking about. And I know the next talk is going to go into it even more. We're getting more and more into targeted delivery to the nasal cavity. So that is driving change in nasal product testing as well as development. Targeting regions like the olfactory nerve and the nault region require new tools that provide an indication of performance as used by the intended patient, kind of what we've been talking about all along here. Human realistic qualitative nasal cast studies are the first step in accelerating your product development. This takes into account the human variables. It helps with device formulation screening, and it is targeted delivery optimization assistance. With this, the device orientation can be very important. It can affect the pump delivery, the spray pattern, the droplet size distribution, as well as deposition, depending on the device that you are using. There are some devices out there that no matter the orientation, they will react the same way, but not all do. So it's something you need to look at in your development and make sure that you're proving. So local nasal therapy, systemic drug delivery, central nervous system delivery, and vaccine nasal delivery are all things that are being looked at right now for that targeted nasal drug delivery. In a human, idealized nasal cavity models represent several important areas. So you have your nasal vestibule, which is really not therapeutic, and a lot of times the product will end up just dripping right back out of the nostril anyway. You have your middle and lower turbinates, your upper turbinates, your olfactory region for potential access to the blood-brain barrier, and the nault area, which is really used for vaccine administration. And then you have your nasopharynx filter, excuse me, in the back, which basically gives you your respirable fraction. So what actually makes it past the nasal cavity. So one of the things that can be very helpful and can advance deposition are nasal cast analysis. This can be a really rapid screening tool to understand the impact of product changes on deposition. This specific example here is done with the Koken model. So this is a completely qualitative, you can look at it. Again, we could do this reverse image here so that you could see the sections that are covered. There are quantitative options out there as well that we have that you can do and go a little bit further using HPLC to see exactly how much is going where. So we use our Proveris by design, which is human focused. Again, we're looking at the human usage because it greatly impacts the deposition in the nasal cavity. This measuring the effect of critical usage factors on deposition can help guide development by identifying the most influential factors to adjust. This can be the actuation profile that we looked at earlier, that automated actuation, the insertion angle, the depth, and of course, how the patient is supposed to breathe and what happens when they don't breathe the way they're supposed to. All of these things should be looked at. This ensures that the product can be used correctly in the hands of the intended patient in the clinical studies and beyond. That really is, I think, the number one thing. With different products we get to test and we do a whole bunch of studies, a product can look perfect in the lab, but you really have to remember how a patient is going to use it. Because just because it works perfect in the lab doesn't mean that it makes sense for somebody in the field. So we really need to concentrate on that as we move these products through. And then this just goes to the weight of evidence approach, everything that we went through here and including more. When you're talking about the agency and what they might be looking for, they really like to see all of the evidence. There are guidances out there, and the guidances will say, do this and it should look like this. But what happens when you do it and it doesn't? Or your product, for some reason, you can't do that. Well, we really push expanding that testing toolbox by incorporating new technologies to traditional testing best practices. And with that, you have all different evidence that you can then put forth to the agency to prove why your product works and why a patient can use it the way that it's intended.